Hello, everyone. My name is Cain, and I'm here to talk about the development of a novel peak detection algorithm for comprehensive two-dimensional gas chromatography. So typical peak detection methods for GC/GC data either rely upon the modification of 1D methods, or they rely upon using a watershed-based image analysis approach. And this latter choice has been popularized by commercial software such as GC Image. However, the problem with a lot of these peak detection algorithms is that they're commonly performed on the total ion current or TIC chromatogram. And so therefore, for analyte signals at low concentrations, the noise from every single mass channel can ultimately obscure those low signals. So therefore, analysts can typically try to use a few mass channels to target those specific analytes that are at the low concentrations. However, this approach only works well for samples that have been well characterized and where the analyst knows exactly what analytes to target. So therefore, we propose a non-targeted peak detection algorithm that will discover low signal peaks using the entire mass spectral dimension. And we term this the enhanced TIC. So our enhanced TIC algorithm first starts with the 1D vectorized form of the GC/GC chromatogram, shown here in step 1. This is an example of just one peak in that 1D form. And then we baseline correct and recenter the data before actually calculating a signal threshold that's applied to every single mass channel. And the signal threshold is based upon the standard deviation of the noise on that mass channel multiplied by 6. And that's indicated by the red line here. We then find the peak maxima that's larger than the threshold on every single mass channel before ultimately trying to determine the window of data to include for each peak maxima. So that way we can ultimately capture the full peak shape. So therefore, signals that are not part of the data window are zeroed out, and then we ultimately sum together all the mass channels to create our enhanced TIC, shown here in step 6. So therefore, we decided to apply our enhanced TIC algorithm to a complex mixture of a yeast cell extract, where yeast cells were grown in glucose and had their metabolites extracted in methanol and triazine buffer. And here I show the example of the total ion current chromatogram for the entire separation. And if we look at a few select mass channels that target the TMS adducts, carbohydrates, and sugar phosphates, we can see that there are analyte signals highlighted by the gold stars that we can see at these mass channels, but their signal intensity is so low that we cannot see them in the total ion current chromatogram. So therefore, we decided to apply our enhanced TIC algorithm to two sections of this yeast cell extract, labeled here as section 1 and section 2. And here I show the chromatograms for section 1 and section 2, where the top 2 chromatograms are for the standard TIC and the bottom 2 chromatograms are for our enhanced TIC. So for the section 1, the standard TIC using a watershed image analysis approach was able to detect 26 peaks, while our enhanced TIC algorithm was able to define 54 peaks in the separation, leading to a 33% increase in the number of peaks detected. As for section 2, the standard TIC was able to find 22 peaks, but our enhanced TIC was able to actually find 36 peaks in that section, leading to a 64% increase in the number of peaks detected. And so something that we decided to do using our enhanced TIC algorithm, since it targets these analytes that are below the limit of detection in the standard TIC, was actually try to understand the true distribution of peak heights in our separation. So here on the right, I show the number of peaks versus the peak height threshold for the two sections of our yeast cell extract chromatogram. And we ultimately have fit these to an exponential distribution to try to determine if that best describes how analyte concentrations act in a complex mixture. We can describe how well the exponential distribution fits to the data using either a lack of fit metric, which should ideally be close to 0, and also an explained variance or R-squared metric, which should ideally be close to 1. So when we look for the two sections of our chromatogram, we see that we do have lack of fits less than 10% and nearly ideal R-squareds, showing that the peak heights in this complex mixture does follow an exponential distribution. Something else that we wanted to do with our enhanced TIC algorithm was evaluate it against statistical overlap theory, or SOT. That theory describes the degree of analyte overlap and the nature of its dependence on peak capacity. And so there's a couple things that we can define. We can define the saturation factor of a chromatogram, which basically describes how crowded is the chromatogram. And then we can also use it to predict the number of peaks predicted in a chromatogram. And so therefore, the number of peaks predicted has an exponential decay as saturation factor or peak crowdedness increases. If we want to be able to compare separations that have different levels of peak crowdedness in the number of peaks they detect, then we can divide the number of peaks predicted by the unit peak capacity to ultimately be able to get the relationship shown here in the bottom graph. And that's going to be the relationship that we use to evaluate our enhanced tick algorithm versus the standard tick algorithm for different chromatographic simulations at different saturation factors. So here I show an example of different simulation at a saturation factor of 0.1 at 4 different signal to noise ratios, 100, 10, 1, and 0.1. And the entire top row of chromatograms is for our standard tick and the number of peaks detected using a watershed image analysis approach. And the bottom row of chromatograms is using our enhanced tick and the number of peaks that it detects as well. And so for this chromatographic simulation at a saturation factor of 0.1, we can see that at a signal to noise ratio of 110, both the standard tick and the enhanced tick perform comparably well. When we get to a signal noise ratio of 1, that's when our enhanced tick starts to outperform our standard tick, since the standard tick was only able to detect 6 peaks in that separation window, while our enhanced tick was able to define 22 peaks in the separation window. And then ultimately, when we get down to a signal noise ratio of 0.1, that is where both our enhanced tick actually starts to fail as well, indicating that our enhanced tick algorithm is ultimately limited by the background noise. Since this was only for one saturation factor, we simulated chromatograms using a variety of saturation factors from 0.1 to 1, and again, looking at those 4 signal to noise ratios, where the black line in all of these graphs indicates that relationship between peaks per peak capacity and a saturation factor predicted by statistical overlap theory. The blue squares are the number of peaks detected per peak capacity for our standard tick. And then likewise, the red squares are also for our enhanced tick. And so what we can see is a signal to noise ratio of 110. Both the enhanced tick and standard tick does predict comparably well as to what the predictions are made by statistical overlap theory. However, when we get to a signal noise ratio of 1, that's when our standard tick has greatly deviated from predictions made by statistical overlap theory, and our enhanced tick starts to deviate as well. And then at a signal noise ratio of 0.1, that is where our enhanced tick starts to deviate from predictions made by statistical overlap theory. Again, we can use a lack of fit metric to describe how well does statistical overlap theory fit to the data for both our standard tick and our enhanced tick. And those values are shown here, where at a signal noise ratio of 100, our standard and enhanced tick have very low lack of fits. And then as we get down to a signal noise ratio of 10, that is when our standard tick starts to deviate in the number of peaks predicted from a statistical overlap theory, while our enhanced tick still performs well. And then at a signal noise ratio of 1 and 0.1, that is where both of our enhanced ticks start to deviate as well. So the low lack of fits at a lower signal to noise ratios ultimately indicates that the enhanced tick provides results that are consistent with predictions made by statistical overlap theory. So in conclusion, we showed the development of a novel peak detection algorithm, and that peaks that were previously obscured by the background noise ultimately have their signals enhanced. 33 to 64% increase in the number of peaks detected for that yeast cell sample. And the use of our enhanced tick algorithm also allows for a deeper understanding of concentration distributions in complex samples, and that the enhanced tick yields results that are consistent with statistical overlap theory at lower signal to noise ratios. With that, I'd like to thank you guys for your attention, and I look forward to giving my 5-minute burst presentation to you virtually. Thank you.